Hi, my name is Scott Berger, and I'm currently a fifth-year PhD student in the biophysics program at Stanford University. I work in the lab of Yogi Kistel. So, bidirectional DNA replication is critical for cells. You start off with one copy of your genome, or more depending on the organism, but ultimately you need to duplicate that in order to make a new cell. And so, cells have figured out ways of duplicating their DNA that include producing not one, but two DNA replicating machines called replisomes. At a particular location, at a particular time, both need to be created at once to ensure that the genome is fully replicated. You can imagine that if there were sites where you only created one DNA replicating machine, one replisome, that they might fire in opposite directions and leave an unreplicated region. And so this would be considered incomplete replication and may result in cell death or even the development of cancers downstream. And so if you want to understand how cells regulate bidirectional replication, you need to understand how cells initiate DNA replication. So during G1 phase, two of the dormant helicases called MCM27 are loaded onto the DNA. Now, only in S phase, you get the recruitment of two more factors, CDC45 and GINs. These essential subunits of the replicative helicase allow it to become activated and recruit all of the other downstream factors, such as the polymerase. And so what I've been doing is using egg extracts to study this exact process. And so in short, you can use the eggs from Xenopus laevis frogs to create an extract that can replicate any DNA, repair any DNA, and is also conserved with humans, and so it is medically relevant. So if you take this extract, you can use antibodies and remove any protein you like. And then we add back a fluorescently labeled version of that protein so we can study it under the microscope. And so here I'm showing you what are known as chymograms of this exact process of DNA replication initiation. So let me walk you through it. Initially, you're looking at nothing. This is just background. And so this chymogram is showing you basically the same region of space imaged over and over again in time. And so the time slices are stacked on top of each other. So in one frame, two copies of GINs are recruited. And then sometime later, usually about 2 minutes, DNA replication begins. And so I then looked to CDC45 to look at how it is recruited. The same story effectively applies, where two copies of CDC45 are recruited at once to a replication origin, and then about 2 minutes later, DNA replication begins. So, in summary, I've shown you that two copies of CDC45 are recruited simultaneously, and the same is true for GINs. But I'll tell you now that neither CDC45 nor GINs dimerize on their own. And so we've posited the existence of a scaffold protein, which is what mediates the recruitment of two copies of these proteins at once. You can imagine that the simultaneous recruitment of two copies of CDC45 and two copies of GINs provides a layer of regulation to ensure bidirectional replication. I'm really grateful to the support of the ARCS Foundation and look forward to continuing this research. Thank you very much.